Thanks for the organizers for giving us this opportunity. We're going to tell you about work in progress. So some of you may be familiar with Aviva Systems Biology. We've been around for quite a while, similar to a number of the research tool companies, the last 20 years developing polyclonal antibodies using peptide 14-mers, and we're actually going to reveal what all those antigens are. We're not interested in holding that information. If somebody wants to copy our portfolio, feel free to go ahead. We've been around quite a while, so over 24,000 self-manufactured polyclonal antibodies that are affinity purified against those 14-mers. They've been in the catalog since 2010. The strategy at that time was to go broad with a focus on transcription factors. At that time, the focus was on chip versus a DNA array or chip on chip. So a nice portfolio. However, as has been discussed over the last 7 years or so, really the market has moved and the requirements are changing. We're really focused now on a new strategy developing recombinant antibodies against some of those same targets. Additionally, we purchased a company in 2017 called Genway Biotech. This is a CRO or CMO that manufactures recombinant proteins. So we leveraged that team in San Diego to build an R&D team in San Diego. I was brought on 2 years ago. So the last year, we've kind of moved the ship towards developing high-quality reagents. And we're going to just give you an update on where we are in that journey. We're based in San Diego. We have about 30 people there, another 30 or so in Beijing and Tianjin, where we still make the polyclonal antibodies. Going forward, we're still taking, I would say, a conservative approach. We do think animals, at least for the research tool market, are the best and fastest way to generate a good, well-characterized antibody. As I said before, those 24,000 manufactured antibodies are complemented by a number of other products that we source from all over the world, but we also have GMP ISO 13485 certification to manufacture recombinant proteins, mostly for diagnostic companies, point of care, immunoassays within the infectious disease areas, everything from HIV to influenza. We do have the ability now to do high throughput antibody characterization. We've invested in our own Characterize instrument that we're leveraging not only for our internal development pipeline, but also for as a service. Both biotech and pharma are coming to us now to evaluate their candidates. We're trying to move that step up early in the process of antibody development to make sure we understand how antibodies are working before we characterize them in the actual applications that our customers are going to use downstream. So as I said before, we have about 24,000 self-manufactured affinity purified polyclonal antibodies. We're trying to move the ship towards a portfolio of replenishable reagents that are highly reproducible and highly characterized. We probably are targeting 7,000 to 8,000 different gene products, mostly in humans, some cross-reactive for mouse. As I said before, we know all the epitopes, at least the antigens that were used to develop these. So in our antibody validation, we've leveraged some of the 5 pillars. Of course, Matthias's favorite in leveraging multiple antibodies to different epitopes. We can do that because we have the portfolio. We're also using other things like SPR, of course, as a member of ICAROS. We're really happy with performance of some of our antibodies in there, and it's allowed us to help weed out some of the antibodies that aren't working. So, of course, we've gone to the market. I think this probably is a CITAB figure, actually. So certainly, we believe that the approach that ICAROS is taking is kind of a fundamental baseline approach. Certainly, it's not going to be relevant for all targets, but we like that approach. It's simple to understand. Our customers understand it, and it gives us confidence in the antibodies that we're working on. So we've launched 1,000 or so antibodies, or relaunched 1,000 or so antibodies that are using genetic validation. Mostly this is in collaboration with Icaros that's validating our antibodies. As I said before, using independent antibody recognition, relative expression in either cell lines or tissues. We've added a fourth. It's not really a pillar, but using the Cartera, which allows us to do high throughput SPR, all the new antibodies that we're developing will have affinity, full kinetic profiles associated with those. What we're doing is developing a database for correlating those affinities with different functional assays that our customers are currently using going forward. And this is what it looks like. Some of the new antibodies we're developing that I'm going to talk to you about now are performing very well in our hands. For example, here's an LCK antibody that we've recently developed. This is still a polyclonal. We have the spleens and B cells to clone out heavy and light chains for this, where we're seeing very good specificity as well as sensitivity for the antibodies that we're developing. Instead of using peptides, we're using either full-length or partial-length proteins, mostly in E. coli, but when needed in either baculovirus, yeast, or mammalian cells. There's a good example of some of the Icaros data that's coming out. I think a number of the antibodies that have been tested, even though they're not renewable at this point, they're still polyclonal antibodies. We supply the market with what I'm calling the long tail of antibodies, where there may not be a recombinant antibody available. In some cases, we have the gold standard, at least according to Icaros. When you look at traditional applications like Western blot, immunoprecipitation, I would say to a much lesser degree, immunocytochemistry or other imaging applications. So we went back to basics, and we're not taking a very sexy approach, but we're putting together a workflow that we've miniaturized and now are attempting to automate. It's very simple using single B-cell cloning. We've scaled antigen development in San Diego with the team there to do this at scale, to go after up to 60 antigens a month. Again, I said these are partial or full-length proteins. We're leveraging some of the new databases that are out there to improve not only antigenicity or responses in animals, but solubility and expression in bacteria and other systems. Again, we're using rabbits. We like rabbits. We've been using rabbits since the beginning. In almost every case with these new proteins that we're developing, we're getting immune responses and B and T cells from at least one of the 4 to 5 rabbits that we're using. And now we're relying on single B cell sorting. So we're doing 6-color sorting in a Sony instrument. It's in San Diego. We're really happy with that instrument. It's working very well. This is not a marketing pitch for instruments, but we put the Cartera downstream of the Sony, and we really like this workflow. So at small scale, we're cloning heavy and light chains from upwards to 96 times 2 plates for each rabbit or each target we're going after. And we're immediately going into cells, doing transient transfections with PCR products before cloning, are able to generate full kinetic profiles against all of those candidates using the Cartera LSA, and I'll talk a little bit about that next. So target selection, of course, we're doing it the old-fashioned way. We don't have a crystal ball. We read the literature, we use the databases, we look at what the competition is doing. We're looking at publications. These are our top 10, for example, current polyclonal antibodies that we're going after. This is just an example. Feel free to take these. We'd love for you to copy ours. These are not the top 10 for everybody. And I think this is really related to the history of Aviva, the targets that we initially went after, and the availability of other antibodies in the market to some of these targets. So we know the structure now because we're looking at things like AlphaFold. So we've actually automated our antigen selection, leveraging a number of different parameters, using different weighting scores to select different parts of these full-length proteins to express it in the platform that we're interested in. So AlphaFold, I think, is a very powerful tool. I think others in this room are using it as well. It has informed us on what the best parts of a protein might be useful for making a stable soluble protein instead of relying simply on primary sequences. So we've done this for the whole human and mouse proteomes, so we know what we're going after. We're using some of the traditional approaches, some of the algorithms that are out there, but the way we've weighted them, including structural information, I think is a little bit different. So this is kind of our own proprietary workflow that we've developed over the last year. And based on that, we've gone into over 500 targets, right? So thousands of rabbits. Right now, almost in every case, we're getting immune responses in the process of evaluating the serum, B cells, and spleen cells that come out of that. And these are some of the polyclonals that we've developed more recently. These targets are in the pipeline for recombinant antibody development as well. When I first came on board, I would have loved to have taken the 24,000 products we currently have. Unfortunately, the blood was not available for those. We had precipitated serum from them. It's really hard to do at scale protein sequencing or mass spec analysis to try to recreate that antibody. It can be done, but I think the best way going forward is combining that with genetic information. So you need the DNA sequence really to do this at scale. And so that's why we're actually going back to the beginning, but leveraging our understanding of what peptides worked previously at Aviva for generating a good or specific immune response. So to get to the meat of this, we're leveraging the Cartera for our internal pipeline to go from this polyclonal to recombinant portfolio. A single B cell cloning is really the most straightforward approach. We're still leveraging the power of the animal. We are getting full kinetic profiles plus epitope binning to help generate fit-for-purpose antibodies in the applications that our customers truly are using and desire. The Cartera allows us to do things in replicate, so you really couldn't do SPR at scale previous to this instrument's launching. This allows us to actually do things in duplicate or triplicate, so if you get a bad well or a bad location on a chip, you can quickly throw that data out. Software is very user-friendly, and it's a quick turnaround, both for our internal customers in R&D as well as our external customers. Lena mentioned this, and I think some of the other speakers as well. We think it's important to understand the kinetic profile of every antibody that you're going to launch. I know Abcam back in the day has evaluated a number of their epitomics antibodies and shown those to be of high value and high affinity. Mark Craig published last year now, I think, a really important paper showing that at least if you're developing an agonist, which I think there is a role for tool antibodies to develop reagents which actually act as agonists, not just antagonists. It's really important to understand the kinetic profile. So clearly the highest binders, at least in the context that he was looking at, this was shown both in vitro and in some mouse models in vivo. It's really important to understand the KD and especially the off rate. So the differences between an agonist and an antagonist in the models that they were looking at were really related to the off rate. And so we have some good examples where we're developing tool antibodies where in a single campaign we're identifying candidates that have very different off rates but the same on rates. And ultimately, yes, you get a different affinity, but understanding those I think will be important to select antibodies that are fit for purpose down the road. So the Cartera is nice. The sweet spot really is doing many ligands or many antibodies in this case against a single antigen. That's the best case scenario. So you can go up to thousands of antibodies a week at this point. We tend to do 96 to 384 if it's just kinetics. As I said before, fast turnaround is the key, but also the low amount of material required to do this. So you only need roughly 200 nanograms of antibodies and picomoles of antigen. So you can actually develop these at scale to evaluate your targets when you're going through a campaign. There's excellent regeneration protocols to do the competition assays that are required to do epitope binning. The data integration is fairly easy with the software that they have there now, and there's a number of different chips you can use. So we've leveraged this not only for antibodies, but a bunch of different formats of those antibodies, but also even things like small molecule libraries, for example, that are attached to DNA. So it allows you to do kinetic evaluation of larger sets of candidates than you'd ever been able to do before with a different platform. And here's some of the initial data that kind of gave us confidence to invest in an instrument like this. So as I said before, we're a polyclonal antibody. The first thing we did was just take 48 of those polyclonal antibodies in our portfolio and test it against 48 different peptides or proteins that we have access to. It was clear that at least for these antibodies that we tested, at least for these antigens we tested, we had very specific antibodies in our portfolio that were giving us very good KDs when measured. The most interesting thing for us is that we could actually measure polyclonal antibody on rates and off rates, and that there is a one-to-one fit. And we think this is due to the fact that we're actually purifying these polyclonal antibodies against the antigen, so against 14 mers. So they're polyclonal, but at the same time, they seem to be acting like a monoclonal, at least for that antigen that we're looking at right there. So in looking at some important cancer biomarkers, some signaling proteins that a number of people are looking at, we're really interested in RAS and developing additional reagents around RAS. We have really nice antibodies, at least based on some peptide and protein in vitro data. When we look at this is that same KRAS protein, looking at its binding to actual full-length KRAS, we're getting the same kinds of results. The upper left corner in this case is actually detecting His. So this is a His tag protein, so that's our control in every experiment where we have His. The ones on the bottom are other KRAS antibodies that we've had access to. So this gave us the confidence that going forward, instead of using this to evaluate this polyclonal portfolio, we want to leverage this going forward to make recombinant antibodies at scale for important targets like KRAS and Notch. One of the things we did, though, as part of that is look at some of the traditional applications that customers are using, including Western blot in both cell lines and tissue, and looking to see if we can see any correlations between some of the affinity data that we're generating and some of the application data that we have or we're generating for those same targets. So we're creating this large database, which is going to have affinity profiles for all of the current antibodies that we have, determining whether we can say anything about the performance of those antibodies relative to a specific application, and if we see differences in the kinetic profile. So a particular case study is related to a target that I can't tell you kind of the identity of it, but I'll tell you the nature of it. If you think about the histone code, it's really important to understand kind of how antibodies are interacting with parts of that histone code. So we wanted to make antibodies against some tool reagents that are targeting different PTMs. So we immunized rabbits using our single B-cell cloning workflow with an antigen that contains two PTMs, which are roughly 3 to 4 amino acids away from each other. We were able to generate hundreds of clones based on this, and then we challenged those clones in small scale with peptides that either have the first PTM, the second PTM, or both PTMs or neither PTM. And we were startled to find a large collection of antibodies that are behaving very differently depending on what antigen you're giving them. So in this case, when challenged with the first PTM, these are actually duplicates separated by one well here. We're identifying candidates that recognize the first PTM only or the second PTM only, or dependencies on the first or second PTM, or in combination with those PTMs, and they have very different kinetic profiles. For example, here's the second PTM. What you see are the same on rates, but very different off rates here, and they're different candidates here. The last one is combining the first PTM and the second PTM together. This could be histone or acetylation or vice versa. What you can say is that there are other candidates that are coming up that are giving us very stable and high affinity KDs. When you overlay those, you can clearly see that we're getting a pool of different antibodies that are recognizing these epitopes with different on rates and different off rates. Right now, we're characterizing these in typical applications that customers would use to determine how we might commercialize these as tool reagents to go after something like the histone code. I'll quickly go through some of these last ones to ensure that we have time for questions, but we have this single campaign. We're able to identify antibodies that are either independent of the two PTMs or dependent of the two PTMs or different combinations of those. And I think that traditionally it's been very difficult to do this with ELISA at scale, to go after this many candidates at once to ensure that you're capturing the full repertoire of candidates that are coming out of the campaign. I'll take 2 minutes to talk about epitope binning, because for every target we're going after, we're also doing epitope binning. This was mentioned previously in a number of the other talks. I think it's important not only for therapeutic antibody development, potentially for tool development as well, specifically immunoassay development, which is why we're leveraging it. It's very easy to do this. So the first binding event that takes place, if you're doing this in 96 versus 96, is shown here, for example, in the first sensogram on the left. You basically add another antibody to that same well or chip, and this is fully automated. And if the signal goes above, it means you're potentially addressing a different epitope. If the signal stays the same or goes down, you're potentially addressing the same epitope. And based on that, you can quickly develop heat maps, which define bins, which allow you to identify potential antibody pairs, but also potentially antibodies that are interacting with different epitopes that may be important for different applications, whether it's therapeutic, diagnostic, or as a tool reagent. And you can express those and show those in many different ways to select the candidate that you want to go after for your downstream applications, whether it's a dendrogram or communities and plots. And we're doing this now for all of our internal antibody development. It's very easy to do. It's fully automated. As long as you keep it under 100 antibodies at a time, once it gets above that, it becomes very difficult to deconvolute that 96, for example, 96 competition assay. So the next 18 months, we have about 500 targets in the pipeline. We're developing antibodies against clinically relevant biomarkers and diseases. We're working with collaborators. We have line of sight to do 60 targets a month. We think it's truly important to move away from polyclonal antibodies. Will always have their role, but as Icaros has shown, you are able to generate recombinant antibodies for almost every target that they've evaluated so far, and there's no reason for us to, it's a headache for the customers, but it's also a headache for the manufacturers to have to rely on polyclonal and rabbits or animals to manufacture these going forward. So one last slide. We don't just do antibodies. So the Cartera, it's not an advertisement for Cartera, but allows you to do not only antibodies, but other things. So we're using small molecule libraries attached to DNA, for example, and you can do this in many different formats, and we've done that already. So I'll stop there. Feel free to reach out to me. I'll be here the next day and a half, and I'm happy to hook up via email or Zoom or whatnot. So thank you.